Welcome Life Science learners to another installment of Life Sciences. Trust that you guys are well and excited about our lesson today. In our lesson, we're going to be focusing on the revision of genetics, genetic engineering, and a very important skill, writing essays. So I trust that you guys are excited and looking forward to our lesson. Let's get into genetic engineering, let's unpack some terms, and let's look at how we apply these terms in the context of a few questions. It's important that we recognize that this section is based on concepts that are derived on terminology. So it's important that you write these terms, guys, and have a list of these terms that you quickly refer to when revising or preparing for a section on this test. So let's move on to some core concepts that we're going to look at. We're going to look at the overview process of genetic engineering, and as I mentioned, we're going to unpack some concepts called revision on genetic engineering. So guys, it's important that when we look at this, the aspects of the section on genetic engineering are important. This is essentially a breakdown of the concepts that you need to have when looking at genetics and genetic engineering. We need to know all the terms around genetic terminology, and these include the terms alleles, locus, homozygous, heterozygous, dominant, recessive. Remember those. We've got to know patterns of inheritance, and these patterns are complete dominance, incomplete dominance, as well as codominance. The pattern in which characteristics which are inherited on the gonosomes we refer to as the inheritance of sex-linked characteristics. So we've got to know how to determine gender, which is in the inheritance of sex, but also the concept of how characteristics are passed down when they're associated with the X chromosome. So we refer to that as the X-linked characteristics or the sex-linked characteristics. The next aspect would be the concept of monohybrid and dihybrid crosses. It's important that you know how to lay out a genetic diagram that shows you a monohybrid cross, as well as being able to interpret the Punnett square in a dihybrid cross. The concept of mutations such as sickle cell anemia, hemophilia, color blindness, and other abnormalities, and understand how these are inherited by using images or charts called pedigree charts. We need to know the concepts of genetic engineering and the different processes that occur, examples of which are cloning, recombinant DNA technology, somatic cell nuclear transfer, and even a new concept called CRISPR-Cas9, and so that's a gene editing tool. It's important that you understand that genetic counseling relies on information that you assess from pedigree charts as well as from genetic screening. The idea of paternity testing using DNA fingerprinting and blood groups is another aspect that you would be required to know when we're revising this section. So let's look at some terms related specifically to genetic engineering. Guys, the concept of genetic engineering is being able to engineer, change, make an organism that is genetically different, and that is either to the benefit of the organism or to the benefit of the person that is manipulating them. In this case, it would be the genetic engineer. The concept of using living organisms, biotechnology, using living organisms to produce useful products, and that comes from using bacteria, fungi, and viruses to produce the likes of vaccines, in baking, as well as using bacteria to produce things like human insulin or growth hormones. We got to know what genetically modified organisms are and the concept of GMOs, the concept of polyploidy and how organisms have multiple sets of chromosomes as a result of genetic engineering processes. The idea of using genes to treat certain conditions we refer to as gene therapy. And finally, the concept of cloning and how that is used in the context of creating asexually genetically identical individuals. The next concept which we use in cloning is called somatic cell nuclear transfer. It's essentially transferring the nucleus from a somatic cell into another cell. The technique of recombinant technology essentially points to how we recombine genes from one species into another, and that process creates genetically modified individuals. The concept of what a plasmid is as a vector, and how that is used to transfer genetic information. And what happens at the end of recombinant DNA technology is we have a recombinant DNA. There are enzymes that are involved in the process, such as ligase and a host of restriction enzymes. And these are tools that are used to cut the DNA and to stick them together. The next concept, transgenic technology, essentially transferring genes into another species or individual, and that creates organisms that are transgenic. The idea of selective breeding or artificial selection is where an animal breeder chooses individuals with specific characteristics and crosses them. And that's also seen as genetic engineering at a very high level, where we're selecting characteristics, also referred to as artificial selection. The concept of using vectors to transfer genetic information, and how individuals that are created show hybrid vigor, generally are having more greater variation or characteristics that are useful for a specific purpose. And finally, as I mentioned, the concept of CRISPR-Cas9, which is essentially a very advanced and new technology in using of gene editing. So that's basically the ability to be able to use a combination of enzymes to edit out certain mutations or to change the genetic makeup of an individual. So as we have done these terms, let's kind of practice this with a set of descriptions and establish these terms. So the first question or description is a way of treating a disease by inserting new genetic material into a patient's cell. And that process is called gene therapy. So when we use individuals' genetic makeup and transfer that into an individual to treat a patient, we call that a therapy using genes. The next description, males and females with desired characteristics are chosen for interbreeding. Again, guys, that is selection of individuals, and we often refer to that as artificial selection or selective breeding, where we're choosing individuals with characteristics and crossing these. The next description, DNA formed by combining a segment of DNA from one source into the DNA of a vector. So that concept is combining genetic information into a vector is called recombinant DNA technology. Enzymes used to glue DNA pieces together, again, that is an enzyme which we refer to as ligase, and it's used basically to connect a recombinant DNA into a plasmid. Mating of organisms not so closely related, again, this encourages what we refer to as hybrid vigor, and that is referred to as outbreeding, where we try and bring in variation by crossing individuals that are not closely related, and that is called outbreeding. An organism with a foreign gene, and that's essentially an individual that we refer to as a genetically modified organism, or in some individuals could refer to as a transgenic organism. So these individuals have got genetic information transferred into them. The altering of the genome by a technological process, and that is called gene editing or genetic modification. And finally, the use of plants, animals, microbes to create useful products is the use of living organisms, and that is biotechnology. So the process of using living organisms to create useful products is called biotechnology. Guys, that's a wrap for this segment. We've looked at some terms, we've looked at some important processes, we've tried to attempt a few descriptions and find these out. I trust that you guys have enjoyed this segment. Have a little comfort break, stretch a bit, get some oxygen into those muscles, and I'll see you in a bit. Welcome back, life science learners. We're continuing with our revision on genetic engineering. Let's get into some application of those concepts by attempting to answer a few questions. Remember that you can engage with me by pulling out a textbook, pulling out a notebook, and trying to attempt some of these questions as we work through them. So let's get into the first question on a process in genetic engineering. So the question reads, the diagram below, which I've indicated up here, shows a genetic engineering process. A donor cell was taken from the muscle cell of a male champion horse, a stallion, to create a new offspring. Guys, this is a technique that is being used in breeding racehorses. It's been a technique used in breeding even cattle. And so when we find that there's a cattle that has, or sheep, or especially in the breeding of cows and bulls that have the desired characteristics of meat and milk production, a farmer will want to create these individuals that have the exact same characteristics. So let's look at this technique and the question. So here's the image, and I've enlarged that for you. So it shows you that a cell has been taken from a champion horse. And so this is a process of being able to extract a somatic cell that has all the genetic information of this particular champion horse. The nucleus of that somatic cell has been removed, and that nucleus is important because it has all the genetic information. Remember that if you want to collect the genetic information of this stallion horse, you need to take a somatic cell. What is unique about a somatic cell is that it has the full diploid set of genetic information, meaning that unlike a sperm or an egg, which has half the genetic information, we've got a cell that has all the genetic information of exactly what this stallion has. So that has been extracted. The nucleus of that has been removed. The nucleus of that is then placed into the ovum of a mare. A mare is obviously a female horse that has donated the egg cell. So the next part of this process shows you the ovum of a female horse has been extracted. The nucleus of the ovum is removed and discarded. So this nucleus is removed. So what has happened is, in this part of the process, we find that we've got a donor for an egg. That egg cell has been stripped of its identity, meaning that we remove the nucleus and we discard the nucleus, which means all we have is just the outer membrane of the egg cell. Into that outer membrane, the nucleus of that donor somatic cell has been inserted. So we now have the ovum being now fertilized with the nucleus of a somatic cell. And this all happens in vitro. So this is happening outside the body. And so this is done, a small electrical charge is given, and that stimulates the ovum to start dividing. It divides in the lab in a structure called a petri dish until it becomes a ball of cells, which often is at a stage of around 7 days, a bit more, which we often refer to as the blastocyst, depending on the gestational period, the period that it takes for the development of that egg. This blastocyst is then inserted into the uterus of the individual that donated the egg, in this case, the female. And so that's the process we refer to as cloning. So let's try and unpack what happens. After a period of time, this female will give birth to a mare, an offspring, that has the genetic information of the individual that donated the somatic cell. And we refer to this process as cloning, and the technique that has been used is called somatic cell nuclear transfer. And so we use the acronym SCNT, which points to how the nucleus has been transferred. So let's get to some questions based on this. Name the genetic engineering process shown in the diagram above. So guys, as I mentioned, here we're producing an exact copy of that individual using a single parent. So this is the ovum, that the parent that donated, and she produces an offspring with the genetic information of one individual. And that process is essentially called cloning. And if you recollect, you've probably been exposed to the concept of cloning, where we refer to cloning in class when we use the example of Dolly the sheep being the first individual that was successfully cloned. The next question, the process that produced Ovum B. Again guys, this process that produces Ovum B, remember that that's an egg cell, and the process that produces that is oogenesis, right? So that process is oogenesis, where an egg cell is produced using oogenesis. There are a few other terms that we can use to describe that, including gametogenesis or meiosis. So these are again processes involved in the production of gametes, in this case the ovum. The next question: why is the donor cell extracted from a champion horse? So guys, as I mentioned, again, animal breeders that choose individuals choose them for desired characteristics. Often during sexual reproduction, we have individuals having a combination of characteristics from two parents. Cloning allows us to be able to use a single individual, in this case, and produce an exact identical copy of that with no variation. And that is the key to being able to clone individuals, is that you're able to produce identical copies, in this case of the stallion, having the desired characteristics of performance, muscle, what is needed to become a stallion or a champion horse in it. And so that's the rationale for doing that. So the donor horse that has the desired characteristic that needs to be copied, and that to be present in the next generation. And that is why we choose those desired characteristics in that individual and we clone that individual. The next question: explain why only the nucleus of the donor cell is used. And so guys, again here was the somatic cell that was taken, and it was just the nucleus that was removed, and only the nucleus. As I mentioned earlier on, in our nucleus we have all the genetic information. So being able to extract the nucleus from this cell and use the nucleus means that we have all the individual's genetic makeup. So we have the full genome of that individual, meaning that cell has all the genetic information to create another copy of that individual. So we say that the nucleus contains the genetic information or the hereditary characteristics in their chromosomes of the champion horse, which we then pass on to the clone when we insert that into the nucleus, into the egg cell donor. Okay, very quickly let's go through the next question. A somatic cell in a horse contains 64 chromosomes. So a bit of math. How many chromosomes would be present in structure A? So guys, what's important is that we recognize that a somatic cell is a cell that is a diploid cell. Again, the context of diploid meaning having a full set of information. So this horse has a diploid set of 64 chromosomes. It's important that we recognize that the egg cell and the sperm cell should each contain, let's divide 64 by 2, and that's testing my maths, 32. So I'm going to retract and say that these cells each contain 32 chromosomes. And so what happens next is that we obviously now need to use that to answer the next bit of this question. Structure A. Let's look at structure A, guys. So that's a somatic cell. And if a somatic cell is removed, it should be a diploid cell, and it would have a full set of genetic information. So that would be a cell that has 64, in this case, sets of chromosomes or chromosomes in them. The next cell that they point to is Ovum B. And Ovum B, again, is from a horse, same species, but again, an ovum is a cell that has half the full set of genetic information. Because it is a haploid cell, we know that it should have half that number. In this case, it would have 32 chromosomes. The next question would be a muscle cell in organism D. In this case, the muscle cell that we point to in this organism D is a fully developed organism having diploid cells. A muscle cell is not a sperm or an egg, which we refer to as gametes. So this is a cell that has a full set of genetic information. So it would mean that it would have a full complement of 64 chromosomes in them. That's an interesting question as well, as I've indicated the answers here. Explain why the ovum labeled C cannot be considered a gamete. So guys, when we look at the ovum labeled C there, we know that the egg cell initially had its nucleus removed. So we remove the nucleus, and what we have left is the membrane of this egg. Into that, we've taken the somatic cell's nucleus and we've inserted that into it. So it's interesting that although it has a full set of genetic information, it is not referred to as a what we call a gamete cell. Let's see why. A gamete, as I explained, is generally a cell that is haploid. However, this ovum, as I mentioned, contains a diploid number of chromosomes because that has received the somatic cell. And if we go back to that, we said that that is a cell that is diploid, and when that information is put in here, this egg cell now has a diploid set of information and hence cannot be considered to be a gamete. Cool, interesting question. Right, so the next bit, and we'll try and get through this question as soon as we read the information and we try and interpret what is illustrated in this diagram. Synthetic insulin is used to treat diabetes and is produced by genetic engineering technology. The diagram represents the process. Let's look at the diagram in a bit more detail. So here we find a human cell, and that human cell contains the full genetic information. From that human cell, the gene that produces insulin has been identified and it has been cut out, and that gene is then isolated. So we refer to that as gene isolation. Cool. We know that in order for us to be able to produce the gene that codes for insulin, we need to put that into what we call genetically modified bacteria. In this case, we find a bacterium whose genome is well understood, that has a plasmid. That plasmid is removed, as you see here. It's spliced open using enzymes, and then we insert the gene for insulin producing protein into that. We now have what we called a recombinant DNA plasmid. That is then inserted into the bacteria, which becomes genetically engineered or modified. That bacteria is placed into a fermentation tank which has all the necessary conditions for it to be able to reproduce, the nutrients for it to be able to grow. Given the right conditions, these bacteria will multiply and produce human insulin. So again, a rapid question that we can take before we wrap this segment up. Define genetic engineering. Again, genetic engineering is a process that is involved in altering the genetic makeup of an organism so that humans or the organism may benefit. The next part to this question would be, describe the steps involved in producing a recombinant DNA. Guys, there are several steps involved in that. I've illustrated that here. As we wrap this up, we can go through these simple steps to illustrate the technology. So first, the isolated DNA gene has been isolated and extracted. We cut out the DNA with enzymes. We then cut open the plasmid, as you would see here, and we use enzymes to do that. We then insert the gene into the plasmid, which we saw, we did that there, and we seal that up using enzymes. This plasmid is then inserted back into the bacteria, and as we mentioned, the bacteria is then allowed to divide and undergo division and growth, and finally, in that recombinant bacteria, we have the protein insulin produced, and that is then purified and packed for human consumption. Welcome back, life science learners. Trust that you've enjoyed the break. Let's get back into the lesson. In our lesson, we're going to continue looking at the skills required to write an essay in life sciences. So, how to answer an essay question? Guys, remember that the essay question is a question that requires you to be able to assimilate information from various different sections. Often we get learners frantic about the essay question, thinking that there's a lot of information that they have to write on a specific topic. Let's look at the essay topic, let's look at how we unpack that, and let's look at the skills required to write this essay. As I mentioned, that the essay in the final examination is allocated a total of 20 marks. To answer this question, it requires that we plan very carefully. Let's look at the planning steps using the following essay question. So here's a question that is taken from a previous exam. The question reads, it's often question 4, and that's in section C. Towards the end of the paper, this is the question that you're often left with. I generally remind learners to keep around 30 minutes for this last question. Again, let's read this. Sometimes the paternity of a son or a daughter is disputed. Let's read further. Describe sex determination in humans and explain how blood grouping and DNA profiling are used in paternity testing. So guys, as you pick this up, there's one component there. We've got to describe sex determination in humans. Second is explain how blood grouping, and third, DNA are used in paternity testing. So it's important that when we read the topic, we're able to understand and interpret the different aspects of that. And as I've done, you would see that I've broken this up into 3 components: sex determination, explaining how blood grouping is used to determine paternity, as well as using DNA to determine paternity. An important note is that no marks will be allowed or awarded for answers in the form of a table, flow charts, or diagrams. And guys, I know that often we tend to rush through, and towards the end of the exam, we want to rush through and write out an essay in the form of a flow chart or simply listing some points. Unfortunately, that's not going to get you the marks. So let's look at how to write this essay after we've planned. So the first step, as I mentioned, is plan and write an essay. Step 1, read the essay questions thoroughly, as I've done, to determine the topic that is being covered. Underline the keywords in the essay question that provide clues to the different topics. And as you noted, I underlined the different parts, and we investigated that this topic had 3 different components to it. The next part would be interpret and analyze the essay question. Identify the aspects of the process required for each topic, and we identified 3 aspects. You may need to read the question once more to enable you to do this. And as I mentioned, if you read this the second time, it helps you to be able to be clear in your thinking around what specifically is required on this topic. Write the first draft of your essay in a logical and organized manner, linking each aspect that is discussed. So I often encourage the learners to have a very quick draft that you write up, and write it as you start your question. Let that be an overview of your planning. Put some ideas around each concept into that part of the draft. This should help you to obtain a higher mark from the 3 marks allocated for the synthesis of your essay. It's important that you recognize that there's 3 marks allocated for the synthesis. And if you think about synthesis is often the development of your thinking around a topic. So here, how would you synthesize the determination of using of sex determination in humans? So you've got to think together and synthesize how this happens in terms of lay out the X and Y chromosomes, lay out the X and X chromosomes in female, and show and describe that process of how sex determination happens. So it's important that in being able to plan that the synthesis does occur. The third step is, in your plan or your draft of the essay, you may take the form of a flow chart. It's okay in your plan to be able to put together some concepts and link them with arrows and ideas, so that when you are going to write that essay down, that you're able to link the concepts together. And that is the value of being able to plan an essay. But you got to note that the final answer cannot be in the form of a flow diagram. So you've got to ensure that you've got sufficient time to now be able to ink that into a paragraph or paragraphs having different ideas, concepts synthesized in each paragraph. A mind map is a useful way to brainstorm the ideas in your planning. It then becomes easy to structure your essay, and it's easier to organize all of that information logically. So I've given you an overview of what a simple mind map could be. So as I discussed this, we recognize that there's paternity testing. We put together the one concept as sex determination. We then had to determine how blood group is used in paternity testing, and then finally how DNA profiling is used. So again, a simple mind map that has illustrated the different components in this essay. The third or the fourth step is write out the final version of your essay. It's important that you put a line across the plan of your essay only if you've written the essay. So often you do an elaborate plan, but you now haven't got to writing that essay. I would then caution you against not putting a line across that. If that's all the planning that you've done, and if you've not been able to get to writing the essay, well, I suppose leave the plan without scratching that off. It's important that if you do write an essay, that you strike out the plan and that you let the reader or the examiner know that that's your final essay. Finally, you read the question again one more time, check if the answer corresponds to the question. As you do that, it gives you a context to what you've left out, what you've missed out in terms of that. Often try and leave some space between a paragraph so that you can go in and add some bit of more information. Proofread your essay carefully. This is the opportunity for you to pick out spelling mistakes, incomplete words, sentences, or ideas. And that often is linked to the time that you have in completing this essay. So guys, it's important that you know that break down the content, synthesize in the plan, and then proofread to see that you've completed an essay with the main concepts in it. I trust that we run out of time in these essays, but I know that in the planning that you do, in the logical thinking that you do, you're able to synthesize a complete essay having all the content that you need in a very pressurized situation. Practice these. Let's have a little break. When we get back, we're going to look at a few more questions and how we unpack that using this concept of unpacking and planning an essay. Short break, and then I'll see you in a bit. Welcome back, life science learners. Guys, we've been looking at revising a very important concept or property in an exam, the writing of an essay. It's crucial that you understand how to write an essay. We've just looked at how to write an essay. We've looked at an example. We discussed the importance of reading that essay carefully. We then said it was important for us to be able to then unpack the different concepts. We then looked at planning, and a plan was an overview of the different aspects of that essay. Put that down. We then went on to saying, well, now put your thoughts together and synthesize that. Now we've done all of that. Let's see if we can apply that to a few questions. So what I've done is I've got a few questions, and we're going to attempt to read through these questions, do an overview of planning, and see how we can construct an essay together. I trust that you will take down these topics and try and attempt writing these essays once you've revised the section. So let's look at our first topic, try and unpack exactly how to look at writing this essay. So the first question, okay, describe the structure of RNA in a cell and the involvement of different types of RNA in protein synthesis. Again, the content is 17 marks. The synthesis is 3 marks, and that's 20 marks. Before we get into the essay, guys, often I get learners asking me, so but how much of time do we need to spend on writing an essay? And guys, essentially if you go back and look at the total of marks that are allocated to any of these papers. It's often a minute and a half per mark. So guys, more or less, you need to keep approximately around 30 minutes to be able to plan as well as write this essay. So as I said, it's important that we read the topic, and I've read that once. I'm going to read that a second time so that I have a clear understanding of the different aspects so that we can unpack that in the essay. So the first word is describe, guys. So that's a description. So we've got to describe. So it's important that we understand what describe means. The structure of RNA. So we've got to describe RNA structure. I need you guys to note how we unpack this topic. So that's the first component in a cell, and the involvement of the different types of RNA. So we then got to describe the involvement of different types. So it's how different RNA is used in protein synthesis. So we've got to look at the use of different RNA in protein synthesis. So guys, I think this is quite an easy topic because it's quite straightforward. There are a few aspects that were easy to unpack from the topic, and I know that for many of you, protein synthesis is not easy, but it's the ability for us to be able to unpack protein synthesis and then explain that in an essay. So now that we've read the topic, we've identified the different aspects, let's look at the next step. So again, as I mentioned, in the first aspect, we've got to be able to very quickly refer to the first aspect. So if I go to the concept here, the structure of RNA, guys. So guys, important for us to recollect our knowledge on the structure of RNA. Remember that we've got essentially DNA, which we've looked at its structure and the types. Now we're looking at RNA and its structure. So you've got to now go through essentially putting together a concept map of just the structure of RNA. So remember that RNA is single-stranded, so you must note that in your planning. It's made up of your nucleotides. Again, it's important to be able to say your A, G, U, and C nucleotides. We got to say that RNA has these sugars, which we call the ribose sugars. It has the phosphate, and if it helps for you to be able to draw this little molecule that shows you that, it does help, guys, and that helps you to again get the context of what you've studied and put that into, again, your planning, guys. This is all in the planning. Include the fact that it has nitrogenous bases, which I mentioned are your adenine, guanine, uracil, and cytosine. Mention the fact that you've got the phosphate group that make up these, that are attached to the ribose sugar here. Mention that the bases are arranged on mRNA in what we refer to as our base triplets, and these are called codons. We also mentioned that on the tRNA, guys, that they are anticodons, and we refer to those, so the concept of being able to include the anticodons there. We also need to discuss the shape of the tRNA, discuss how its structure is, that it's the point of amino acid attachment being there. So what we've done in this part of the aspect of the essay is being able to describe the structure of the RNA as well as the different types of RNA. And as a quick recollection, we've got 3 types of RNA, guys. We've got mRNA, we've got tRNA, and finally we have a very important RNA which we often refer to as ribosome, but that's the rRNA. So those are the 3 different types of RNA that you must be able to elaborate on their structure. And finally, the latter part of the question is describe the role of these RNAs in protein synthesis. And this would require that you now unpack protein synthesis in 2 processes. Remember that in protein synthesis, we have the first step, that is transcription, okay, and the second process that occurs is translation. So guys, this helps me to get my thinking around transcription. I've now got to illustrate the process of transcription. I must mention the fact that mRNA is involved. I must discuss how that is produced, the process of transcription, how the rRNA or the ribosome and the tRNA collectively read the mRNA using the building blocks, which we refer to as amino acids, make what we call proteins. So that description needs to take place. Let's try and unpack possibly how the involvement of these RNAs take place in protein synthesis. We recollect that mRNA forms during transcription. We know that this process is called coding the message from DNA. We describe that the mRNA is produced in the nucleus. It then leaves the nucleus to the nucleopores, and then it moves out into the ribosome. And that process that we've just described now is called transcription. It then moves into translation, where we see the mRNA, which is now that strand containing codons, as we mentioned that. We also mentioned the concept of the tRNA bringing in the amino acids, which we often illustrate using that. The tRNA then brings in the required amino acids. We talk about the bases on the tRNA and the bases on the mRNA forming the codon and the anticodon bonding. The amino acids then come in, and then they held together what we refer to as with peptide bonds, forming eventually a polypeptide chain. And that describes essentially the process of protein synthesis. Guys, these essays are marked very systematically using what we refer to as a rubric. It's important that you understand how this rubric is used. Essentially, it will help you to be able to think clinically around how to structure your essay together. So let's look at the rubric for this particular essay. So there are relevance, logical sequencing, and the comprehensiveness. So those three terms, relevance, so what you've written in this essay was relevant to the aspect. So we needed to discuss the types of RNA, the structure of RNA, and the use of those different types of RNA in protein synthesis. So has the information that you provided relevant to the topic. So you cannot fill space in your essay because you want to take up a lot of paper and impress the reader by filling up pages. That's obviously going to get you into trouble if that information is not relevant. The second part of it is that there has to be a logical sequence. As with the process of protein synthesis, we need to logically and describe how transcription takes place, the process and how that mRNA is produced, and so that refers to the logical layout of cause and effect. So we talk about logical cause and effect. So we say this has to happen first, followed by that. And in the process of protein synthesis, the sequence is very important. And finally, was this essay comprehensive? Did you cover all aspects required, and has they been sufficiently addressed? Further to this, in this essay, the information has to include the structure, the involvement of the different types of RNA, and that there is no irrelevant information. And that gives you the 1 mark for the synthesis. The second mark would be all the information regarding the structure of RNA, involvement of it in the different types of RNA is given in a logical manner. Yes, you've done that, the second mark for synthesis. And finally, we're looking at the third mark for how comprehensive was your answer, in that you had at least 6 to 9 of the facts required for the first aspect of structure of RNA and the types of RNA, and then at least 5 to 8 of the facts required for the process of protein synthesis, where you described transcription and translation. So guys, as you see, this essay can be very, very clinical in the way you write it. It's important that you're able to think logically, keep a cool head, and synthesize that information, knowing that you have all the content that you've prepared. Let's try another essay. The next essay question: using genetically modified organisms as a source of food is supported by some people and rejected by others. So that's the statement that has been given. It's important to read that once more. Again, we know that the concept here is that genetically modified organisms as a source of food has been supported by some individuals and rejected by others. So we're seeing here there's a concept that's being supported and rejected. Good to get that clearly understood early on. The next part, this is the instruction to that. So there's a statement that has been given. Here's the instruction. Explain 6 advantages. Focus on the concept of the number 6. Advantages, guys, of using genetically modified organisms as a source of food, and give 4 disadvantages of using genetically modified organisms as a source of food. So guys, when we unpack this essay, again, the context of genetically modified food is a controversial concept. This essay says that some individuals support them and others are against them. I think if we were to very quickly kind of look at this, you've got to give 6 facts that are for, and you've got to give 4 facts that are against the use. Now guys, remember that this is a 17-mark essay, and you've got to have that at the back of your head, and you've got to have the synthesis concept in your planning. It's important that in this essay we look at what is needed to be able to string together 6 facts that are supporting 4, and again 4 facts that are going against the use. It's important that you have a thorough understanding of the pros and cons of concepts such as genetically modified food, the advantages of them, the disadvantages of them, etc. So there are many topics that we can. So it's important that at this point, as we wrap this segment up, that we often have learners trying to prepare an essay before they write an exam. Unfortunately, you cannot really predict an essay topic, and that's poor planning. I would rather suggest that you spend some time looking at various aspects and seeing how they interconnected. And that's essentially what I've done here. In this slide, I've gone through and I've looked at how, in the concept of life sciences, how different concepts can be linked together. And that's essentially, as I wrap this segment up, I'd like to emphasize that fact, is that in this little blurb that I've put up there, I've showed you that there are various factors that can be interconnected. And that's what I'd like to spend a minute wrapping up this segment. So we know that in meiosis we can discuss the process, the significance of the process of meiosis, as well as how it produces variation. So if you were preparing on concepts around meiosis, know the process, know how it occurs, and give a sense of what happens that produces variation and its importance. The next area is when we look at is DNA and how mutations occur. We know that mutations occur at a gene level as well as at a chromosomal level. Know some examples of gene mutations like sickle cell anemia. Know the way in which gene mutations occur. So we talk about deletion, insertion, substitution. We also know that there are chromosomal mutations. So again, how do these occur? So non-disjunction during meiosis, and talk about some examples. So in terms of Down syndrome, Klinefelter, Turner syndrome. So these are, again, know how they occur, the effects and examples of them. Again, a possible subtopic in an essay. The next factor would be how DNA fingerprinting, profiling, or karyotypes are used in being able to pick up genetic abnormalities, DNA fingerprinting, as well as looking at paternity. So again, the concepts of karyotypes, fingerprints, pedigree charts are important in being able to identify abnormalities, look at inheritance of a specific characteristic. So again, different tools used. The concept of genetic engineering, as you see, has various processes as well as advantages and disadvantages. Know that well. And finally, know some genetic disorders such as Down syndrome, albinism, sickle cell anemia, color blindness, hemophilia, muscular dystrophy. These are important because they often would ask you to describe a few genetic disorders, explain some symptoms of them. And I would encourage you to have a few of these in your notes. These are some of the common ones, but they're often relevant to different sections. Finally, the genetics, as I often emphasize. In genetics, there's a whole lot of terms that are important, and these are fundamental for you to be able to understand concepts and construct an essay. Guys, we've looked at how to plan an essay. We've looked at how to read through an essay. It's important that you take these and apply these and practice these. Try a few essays, and I hope that you've enjoyed the lesson. See you soon. Have a baie lekker day. Cheers.